All right. Welcome to the Craniotomy podcast, where we peer into the minds of those on the front lines of neuromodulation and neurology and psychiatry. Uh, Today, I'll be your host, Dr. David Darrow. Dr. Alex Herman will be joining us shortly, and we are the co-PIs of the Herman Darrow Lab at the University of Minnesota. Thank you for joining us for yet another exciting episode. Today, we have Dr. Jerrold Vitek, who is the Department Chair of Neurology at the University of Minnesota and who leads our deep brain stimulation clinical and research program here at the University of Minnesota. Dr. Vitek has pioneered our modern-day understanding and use of high-frequency deep brain stimulation as much more than a functional lesion. Reading his accolades would take the entire program, and we'd prefer to hear directly from Dr. Vitek. So thank you so much for joining us here today, Dr. Vitek.
Yeah, David, so we can just go on first names, I think. This is informal, right? So Jerry's pretty good, I think. Well, thanks for inviting me. I think I'm looking forward to talking a little bit about, where this all started and where we think it might be going.
One thing I think is really interesting is that in many ways, you've kind of come full circle. You started here at the University of Minnesota in some sense. Would you mind recounting your sort of trajectory to neuroscience and neuromodulation?
No, not at all. In fact, we can start even earlier than that. We can start when I applied to medical school and got turned down 11 times in my first year. Or we can talk about the other 2 years after that I was still turned down and didn't get into my fourth try. But we probably don't want to sit and talk about that all day. But it took me several years to get in. And I never understood colleagues who decided that after one attempt, they were going to stop. Wow. So I did my MD and my PhD here. I mean, the good thing was that when I didn't get into medical school, I went to graduate school. Initially started at a master's program and then went on to a doctoral program. While I was doing that, I kept applying to medical school. When I got in, I'd already spent 3 years in graduate school and decided I was interested in neuroscience and neurophysiology. It kind of kept me in that direction. So I decided to go back and forth. They didn't have an MD, PhD program. So I basically went into the medical school for 2 years, came back then to go and do my PhD, which took several years. And then went back and finished medical school. Actually, I never even did a neurology rotation because I didn't have time. I know it sounds odd, right? I'm a neurologist that didn't do a neurology rotation. I didn't do it. They let me off and said, because you spent so many years in the lab, we'll count it. Although I wasn't doing neuro exams on cats. So, but the bottom line is I got in, went through, finished the PhD, finished the MD in 84, then went and did internship in upstate New York because like an idiot, although I had matched for residency at Hopkins, I had put one internship at University of Minnesota. So I didn't match for internship either. So then I had to find a place to go for internship, and I won't even go through that story. We ended up in upstate New York with a 125-pound bull mastiff and a 4-month-old baby. So I can give you a long story about what that was like, but I won't bother with that either. 1 year there, and then went off to Hopkins, did residency, stayed there for a couple of years with my mentor, Malin Long, which is really kind of the best thing to happen to me because I found a really good mentor. Spent a couple of years in the lab with Malin, and then he took the chair at Emory, and I moved with him. And that's where we started doing a lot of the functional neurosurgery work with Roy McKay, pallidotomies initially, and then DBS after that. Spent 14 years at Emory, and then was recruited to the Cleveland Clinic, again, to develop the neuromodulation program there. At that time, Ollie Rezai and Erwin Montgomery were there. So by the time I got there, Erwin had left, and it was Ollie and myself, and then we built the program. And then I went and came back to Minnesota when the chair job opened up, because being a Minnesotan, I always wanted to get home. It just took me 26 years to get there, right? And so when I got here, there was already some neuromodulation going on, but I think we were able to grow that program with a lot of good people. You guys know we had 3 functional neurosurgeons with Michael, you, David, and Bob McGovern. And now we're expanding off with the excellence that's occurring in the psychiatry program with you, Alex and Alec, among others in your group. So I think with the imaging that's been here and all that, it's been just really nice to have a lot of senior and young investigators coming through the system. And you guys know we've been able to really, I think, build the program. I think we're one of the better programs around right now with the diversity we have. And I don't mean diversity in terms of socioeconomic, I mean diversity in terms of skill sets that people bring to the table. it's a great group. And the other thing is, I think this is the best group I've ever been with, not just from the technical standpoint, but I think also personalities. And the stuff that you guys are doing on the cognitive realm, I thought this would really, really fit perfectly well with the Udall grant. It takes us to another dimension with that work.
And I think one thing that, we wanted to talk about today was what that's sort of built on, which, is really the difficulty with knowing exactly where you are in the human basal ganglia. And I think people don't really understand the nuances of that and how to interpret a lot of the science that has come out of that on the human side. In a primate world, you have a lot more control, but on a human side, it's much trickier to know and confirm where you are, because validation is really, really the challenging part. Can you talk a little bit about, how that has developed over the years in your mind, and how, for example, how we do it is a way that instills a lot of confidence in the rest of the research that we do.
No, I agree. And I was trained by Malin Long, and Malin had done a lot of work in the basal ganglia, non-human primates. So he was at the lab at NIH, I think, in the time of Everts, was well known for motor cortex work. Peter Strick, Tom Thatch. These are giants that were in the field. Tom Thatch and cerebellum. Peter Strick did both basal ganglia and cerebellum. And Malin was put in a lab many, many years ago. He had a paper like in 1970 or 71. But he talked about the different cell types and how they sounded physiologically, firing patterns, how different they were in the basal ganglia. I mean, people didn't know that at all before that time. Yeah. And so he kind of used that understanding to be able to map out where you were in the basal ganglia. Where are you in GPI? Are you in GPE? I'm going through striatum. Am I in motor GPI? I'm in associative territory, limbic. He worked out all those sub-circuits, he and Peter Strick and others, to understand that, there's a different functional organization in that structure. And, if you're in a motor territory and you're either lesioning or stimulating, you're likely to affect motor signs. If you're not in a motor territory, you're not as likely to affect those motor signs. And I think that was pretty critical, basic understanding that I think we kind of forget about it, but it took a long time to develop it. And so when we started looking at doing this functional work, when I was with Malin, the idea there was to use the same electrophysiologic techniques used in the primate for the human. And so my job when we moved to Emory was to set up that equipment. And so I had someone helping me, pull together oscilloscopes. You couldn't just buy it, right? You can buy it now. It's commercial. But so we had a big, we called it Big Blue. It was a big blue rack. We put oscilloscopes in there. We put our amplifiers in there. Everything went into this big blue rack, including the stimulator. And when we roll that into the OR, obviously we wiped it down, right? And we didn't keep it in the monkey lab, but it was made from components like that. And we'd roll that down into the OR, and then we'd use that to record it, and then in the humans. But it wasn't well isolated, so when there was someone in the room next to our OR, and it was actually cardiac surgeons in there. Yeah. And whenever they were opening the chest, we had to stop recording because there was so much noise we couldn't record through it. Yeah. So we'd always know right around 11, 12 o'clock, those guys are going to be opening the chest, and we'd have to wait for, however long it took them to finish.
Oh my gosh.
Wow. So that's kind of where we were back in the 90s, right? Mm. And this has evolved, and the physiology has evolved. We applied the physiology to the human. The first case that we did the mapping, and it was working through the noise problems. It was working through the equipment you had. And our first pallidotomy case that we actually mapped, we weren't sure that, the physiology was going to be the same or that it was going to be organized similarly, right, to the monkey.
Right.
And so it took us from 8 o'clock in the morning on that first pallidotomy that we did until 2 o'clock the following morning. Wow. To work with this. You can imagine this patient, right? This patient wanted to be first. We kept saying, "Are you sure you want to be first?" He said, "Yeah, I want to be first." And he hung with us from 8 in the morning. I mean, you figure that out, right?
He's like healed up by the time you're done.
Yeah, it was interesting. Now we're doing in a couple hours. We can map in a couple hours now usually. So
the hard part that people don't understand is the variance and the heterogeneity between patients. And the way that I have come to understand our microelectrode approaches towards mapping is really algorithmic. While we do have maps, we have an algorithm in our head that says, we're going to be passing through this area, we're going to move into this area, and typically on average we see these features in these other areas that corroborate sort of it's sort of it's a high dimensional map that we're projecting onto what we assume is a very straightforward slice by slice map. Yeah, there is some slice by slice map, but the problem is that there's a lot of variation between patients. Everybody's brains are different. So we're using an algorithmic map projected onto that as our method of continuously validating exactly where we are in their basal ganglia. Some people say, we don't need that algorithm anymore because we just get a scan. But I think what is hard for people to know is what you're alluding to, which is these substructures within even the smallest nuclei in the basal ganglia that may play a major role in sort of directing how we do stimulation and how it affects them overall.
One of the things I think that has really kind of struck me is I won't say the lack of understanding, but I think, we're not trained as physicians in medical school to understand basal ganglia physiology and anatomy and the functional organization. Nobody ever trains us for that.
No doubt.
So you were never expected to know it. I mean, I was fortunate to work with a guy whose major background was working on basal ganglia anatomy and physiology. So it struck me early on with him that, GPE is very different from GPI, but the bottom line is you take a population of cells, they're different. Typically, you go in there and listen to the kind of the physiologic hallmark, right, which is a pausing cell or a bursting cell that you hear in GPE. You get into GPI, typically you don't hear that burster. You can get some cells that pause, but they don't usually pause similar as GPE. It's an art form. I mean, people have tried to do this automatically, right? Develop algorithms to say you're in GPE or in GPI. And I've always found that maybe you can do that, but I think there's enough variation you can't always be sure. So, one thing I always look for is those bursting cells because they're a little unique in GPE. And I figure there's maybe right around 5 millimeters of GPE and maybe a little less, maybe a little bit more.
Right.
I look very hard for a lamina between GPE and GPI. When that quiets down, thing is you can get quiet spots in GPE too, right?
Right. But
you're really looking for that lamina, and then you look for those cells in GPI that are firing a little bit faster, a little bit more tonic, and you look for receptive fields to say, yep, I'm in the motor territory.
Right.
If I'm not finding cells that respond at all, it makes me a little nervous, right? I'm starting to wonder, where am I here? Am I really in the motor territory? So that makes me feel a lot more confident when I start finding cells related to arm or leg. And indeed, contrary to what some people are going to argue, there is a symmetopy in GPI. There's more arm lateral and there's more leg, more medial.
Right.
So you can get a sense of where you are. And, by making that map, you make a couple, 3 passes. I think you need 3 passes to know where you are with confidence. And you want to find optic tract and you want to find capsule. And by finding that, I think you feel a lot more comfortable that I know I'm going to be in this motor territory. And then it's where in the motor territory am I, right?
Right. And that's the sort of patent pending Vitek algorithm. It's that's what you've built in over with all your experience with all those patients that you've mapped. And that algorithm, it is not just a 3-dimensional representation of, an average MRI. If your mom had Parkinson's, how would you want the DBS electrode to put in? Would you want them to spend some time to understand her individual anatomy, or would you want them to sort of guess and hope that, they're within a few millimeters of STN? When you break it down like that, I think I'd want the Vitek algorithm to get us into the right spot with a lot of internal validation that you've built up over the years, and I think that's the part that is actually pretty hard to convey in the literature.
It really is. I had a person ask me once that was, we were out for dinner with a group from industry, and one person was asking me, he said, well, Jerry, just how precise do you really need to be? And I said, well, Tim, if it was your brain, what would you want? And I mean, I think we all know that question, the answer to that question, right? I mean, yeah, you don't want to be dropping down 10, 15 microelectrode tracks. I mean, I get that. But the bottom line is the imaging is very good, particularly at Minnesota. We know, like Noam Harrell and that group with CMRR, are really good. They can get us pretty daggone close.
Right.
But I like to validate, where we are. And we can do that with those 3 microelectrode passes using an even to bend gun.
Right.
And that to me, there are times we still move, right?
Right.
We think we know where we are. When you look at those images and you plan it out, but there are many times yet you're still going to make an adjustment. But to me, having done this for so many years, I don't understand, I guess, how people do this without microelectrode recording.
I think that's a perfect segue to, exactly what a 2 millimeter shift could put you in STN. And, one area that we're really interested in is the sort of non-motor aspects of Parkinson's that, over the years as tremor, as you've had such tremendous success with tremor, now there's a focus on non-motor symptoms because, at least in my limited experience, oftentimes that's a lot of what the families, are really bringing up at sort of the visit with the neurologist as one of the first things that they notice has changed over time for their loved one.
No, you're right. In fact, we had a meeting with, it was put through an organization. We brought a lot of families. So we brought in the Parkinson's patient and their families. And then one portion of this meeting was where we sat down and we put up a list of all the motor problems, right? So we looked at tremor, rigidity, bradykinesia, the gait and balance. And then we asked people what was the biggest concern for them, for the patient and then for the family. You know what they came up with? Things that were not on our list. It was sleep. It was cognition, right? Those were things that were really problems for them because, we can treat a lot of the motor signs other than freezing a gait's another issue. Gait and balance, sometimes based on what it's coming from, you can treat that. Some people respond, gaits respond, but a lot of times it may be inherent posture reflexes that are a problem and those don't respond. Right. But the things that they complained about, they said, look, medication helps us to a large degree. And then of course, surgical therapies are good also for the motor signs. But boy, for cognition, sleep, they had that was their concern. So now we have to focus on the things that, for example, you guys are working on and we can work on together is looking at what does DBS do to cognition? If we can improve motor signs with STEM, why can't we improve some of the cognitive signs?
Right.
Into the right area, the right circuit, do the right stimulation, activate or suppress the right pathways, we should be able to modulate those. Right. I think that's kind of the aim of what you guys are up to right now.
Right. Very exciting frontier. And I think one of the things that, has always struck me is, for example, when we go after subthalamic nucleus as a target for tremor, within just a couple millimeters are the, similar projections that, loops with the huge frontal lobes that we all have, and limbic, limbic regions as well. And it's amazing when you think about it like that we're not affecting those regions even more, even when the DBS is perfect.
I remember a patient years ago, we were first starting to do DBS and we implanted this patient. And sometimes I think when you infringe on kind of getting towards that associative territory, or maybe you're a little deeper and you might be affecting some limbic, remember the fibers are passing through here, right? So we can be sitting in like a neuromotor associative territory, kind of on a border maybe, and there are going to be fibers that are passing around that we're activating. And the question is, are we actually activating some of the associative territories? Or if I end up a little more anterior in STN, and am I activating some motor pathways that run through associative territory? Right. I mean, there's all these questions we have to ask because now we're much more understanding that this is just not about activating or suppressing cells. This is pathways. And so the question then becomes, if we end up a little more anterior, how do we optimize the pathways that we want to optimize? I mean, we have directional leads. Do we stimulate a certain way? I mean, can we activate some fibers and not others based on the orientation of lead? I mean, that's, you guys know that's all the new stuff that's coming up. And I think that really emphasizes our work with biomedical engineers.
Right.
People that can help us model and understand how we're activating these fiber pathways based on the orientation of the lead, the current field, how we stimulate. It gets complicated. It's simple, but it's complicated.
Yeah, and the importance of activating pathways is something we've discovered, also kind of in parallel in DBS for depression, for instance, where we started off going after, gray matter targets and then realized it's really about these fiber pathways that, seem to be mediating the effects that we see. I think we have, on some level, a much more detailed understanding of the anatomy and obviously in movement disorders and Parkinson's disease, but we still don't have, a grasp over how we can be differentially activating different fiber tracks and different pathways to, minimize the potential negative effects of DBS as well as potentially treat some of the actual cognitive and mood sequelae of the disease process itself in Parkinson's.
Yeah, no, I agree, Alex. I think if you remember the early hypotheses, right, where that DBS suppressed everything, and that was what Helen Mayberg used when she was looking at subgenuary 25. Well, bottom line is, from what we know about deep brain stimulation, that early paper we did long ago in 2003, where Taka Hashimoto and my lab demonstrated when you stimulate an STN, you activate GPI. And even though the cells may be suppressed, GPI goes up. So,
right.
We're activating the output. And then McIntyre modeled it. And he said, we're activating axons. So that was some of the first stuff that suggested, boy, that must be what we're doing. And if Helen had thought we were activating, I'm not sure she would have wanted to stimulate in that area, right? But it's probably the pattern. And it's probably, like, again, these power spectrums and oscillations, what are they really doing in the brain? And how are places talking to each other? And how do we change that with activating or suppressing these particular pathways? Now, I was really excited about the stuff, the preliminary data that you and David have gathered in the OR with us. Because when you talk about the motor territory, we're changing things in dorsolateral prefrontal cortex.
Right.
So
right.
I thought that was pretty exciting. And it just validated the fact that I wanted you guys involved in this project, right? Because it really leads us, we can use these Parkinson's patients and we can put things in motor territories and still see that maybe we're affecting some of the cognitive components of this, right? I mean, when you think about it, do you ever make a movement where you're not thinking about it?
Right, exactly.
Sometimes you are. I mean, sometimes it's spontaneous when I'm moving my hands like this as we're talking, but most of the time it's kind of well thought out.
Yeah, and there's an entire theory of how the brain works that essentially supposes that movement and action and thought are all really intricately related, right? It's, related to this idea of embodied cognition, that our brain is really functioning in terms of the potential actions and movements that it can take in any given time. So on some level, I mean, that's pretty abstract, but on some level, it's not so surprising that we're actually finding in these individual patients with DBS and motor regions that there are direct cognitive, consequences, or at least aspects to what we're doing. Because the idea that movement and thought or movement and feeling are these siloed domains, we know that that's not true, right? They evolve, they co-evolve together.
You think about patients that freeze. What do we tell them in the clinic? We tell them to rock side by side, or we give them a visual stimulus, right? We give them a light, say, take this pen light and shine it on the floor and step on it. And sometimes those people, they don't freeze. And there was another group years ago that was talking about visual stimuli for people that had dyskinesias. So, from taking too much dopamine and they start getting all these dyskinesias. There was a person who used to wear glasses that had like red and blue filters on them. And they swore that was suppressing their dyskinesia. So I'm still thinking how that worked, but did it work? I mean, I think if we got to get ourselves out of this box, and Marilyn Long helped me a long time ago. I was pretty rigid about things, like things to be laid out. I like the silos, Alex, but I'm disappointed now it can't be silos. So we have to think outside the silo. But I remember him telling me when we would see odd findings that just didn't fit with the hypothesis. He would look at me and he'd say, Jerry, and in fact, I just talked to him yesterday for my old mentor. He's 83 now, but still crystal clear when I talked to him. And we were reminiscing about what he had told me one time, and I brought him some data that didn't make sense. And he looked at me, he says, Jerry, he said, the data is the data, right? And the point is, it's up to us to look at the data and understand what it might be telling us, not for us to tell the data what it's supposed to be telling us.
So how much, kind of stepping back and thinking back in your career a little bit, how much did you anticipate the directions you would go with it earlier on? And how much was it sort of, maybe not happenstance, but, serendipity and what things worked and what things didn't?
Well, for me, probably first serendipity was just not getting into medical school and not going to grad school, because that drove me along the lines of neurophysiology. And I realized, boy, I really like this.
Mhm.
I liked neuroanatomy. It was my favorite thing to do at medical school, frankly. And then I liked the physiology. And when I initially didn't get in, it drove me down that path. And then I realized I want to do both. So that was one serendipitous thing that happened, right? I initially thought, God, I didn't get in. This is horrible. What am I going to do? And then after that, it all kind of became clear. I know what I want to do. And then I knew where I wanted to go. And I wanted to work with someone like Malin, where I could get that kind of that jump start. Although when I first got to Hopkins, I really didn't intend to do movement disorders. I mean, I didn't intend to do these long 36-hour experiments I've been doing in grad school. I didn't want to be part of that. So I told myself I was going to do neuromuscular, and I want to do neurometrics, EMG, nerve conductions, that kind of thing. And I was walking down the hallway with Dave Kornblath, who was one of my, he was a neuromuscular guy at Hopkins. We're doing consults. And I was telling him, I said, well, I'm really interested in neuromuscular and doing the things that he was doing. And he told me, he directed me away from it, which surprised me. Normally people want you to do what they do, right? But he told me no. He said, I thought I should, he kind of directed me towards movement disorders. And Malin DeLong, who was my mentor again, my first year at Hopkins, would pull me into his office and he'd suggest a project. And I would say, Dr. DeLong, I understand this is an interesting area, but I said, I really don't want to do those 36-hour experiments anymore. And he said, well, Jerry, he's a very calm, nice man, if you ever met Malin. He'd say, well, you just think about it. And he pulled me in his office over the next 2 years about 4 or 5 times. And he kept telling me, well, the work that we do on non-human primates, it's, 4 hours of recording, 5 hours of recording. You don't spend all day. You're not doing 36 hours. And frankly, he wore me down. I gave in because one, I liked him a lot, right? And the work that he did was interesting to me. And so then I agreed that I was going to do a K award with Malin. And that's what drove me into that field. But believe me, it kind of pulled me in kicking and screaming the whole way. But once I got working with him, because of what he did and the kind of guy he was, he got me very interested in the work that he did. And so that kind of pulled me into it. But I never envisioned doing pallidotomies or functional neurosurgery with neurosurgeons and doing mapping and things like that until a guy named Fred Lenz came to Hopkins. And he had worked with Ron Tasker, as you know, is a very well-known functional neurosurgeon in Canada and Toronto. And Fred had developed kind of the microelectrode approach. They were making microelectrodes themselves, right, to do the recording. Not that microelectrode recording hadn't been done; it had been done quite some time ago. But Fred kind of brought that to Hopkins. And then Fred and Malin wanted to develop functional neurosurgery at Hopkins, and then they brought me along because I was in the lab doing mapping into primate thalamus, not even primate thalamus. And so we kind of combined all of that understanding and started doing it together. Because that's how it brought me into that. And honestly, it's almost luck, right? I mean, I didn't intend when I went to medical school to do what I do now. That was never what I planned on doing. I was going to be a physician. That's what I wanted to be. And I wanted to be a neurologist. But then this whole thing led me to Hopkins, led me to Malin. Fred came in, led me to the functional side, brought me into the OR. And then next thing you know, Malin's moving to Emory, and he's recruiting me to go down there. And then it turned out Roy McKay was down there, who had trained at Bojan. And we talked to him about it. And then we talked, well, let's develop something at Emory. And that was my job then, was to develop the equipment. And so I had another guy named Doug Reel, I think his background was physics, and he helped me put things together, and that's how it all started. And then one thing led, once you get into it, right, you guys, once you start it like you guys have now, all of a sudden those paths become a little more clear. At least the early stuff becomes clear. You don't know where you're going to be 10 years from now necessarily, but you do see things that take you down different paths. And then you find that there are certain things you really like. You get intrigued by it. You ask a question. That question leads to another question. And how can I make this better? How do I do the physiology better? How do we know what the best location is? How do we get to that location? How do we verify that we're in that location? When we used to do pallidotomies, we would go the very next day and look at that image and say, where did the lesion end up? And how does that compare to what the mapping that we did, right? So all that stuff fed back into one another, which helped us, I think, to improve. If you don't look at the results of each case, like even now, I've changed my clinic now so that I can go back into the clinic after we've implanted a case, and I can go work with the nurse practitioner and look at the program, because I'm not doing the program directly anymore. I need to understand is, do we make the right decision in the OR when we decide to implant somewhere? And sometimes the proof is in the pudding, right? The proof is in the result. And what kind of monopolar review do we have? How's the patient doing? Those are the things that it's exciting, right? Because it's each one is new to some degree and leads you in a different direction sometimes.
Right.
At least that's how I feel. I think you guys are a perfect example of that, aren't you? Seems to me, Alex, you've taken a very different route in your life, and David, you're too. I mean, I heard your story in mathematics, right? You gave a lecture just recently. I listened to it. Started off in mathematics, and all of a sudden you decided this is not for me. Isn't that how it worked for you?
Yeah, no, definitely. I couldn't have predicted it for sure.
And Alex, your background, I mean, you can talk.
Alex also has like a similar background.
Yeah, my background was in physics, and I thought I was going to go into physics and do theoretical physics. And, I just went with my intuition and feeling at each step. And, as opportunities came up, I, pursued them. I mean, I'm sure those particular things that opened up along the way, they ultimately shaped, where I ended up, and had different things arose. I could have ended up somewhere completely different. Maybe that's true for all of us, and then, later on we tell a story about how it all made sense and all of that. But
looking backwards, it's always easier to explain things than when you try to look forward. Yeah. But, if you look at what your relative skills are, though, in mathematics and physics, isn't this where a lot of this is going with modeling and signaling and all of this?
True, that's for sure.
And I think you guys are well positioned with that. So what do I have to do? Well, I'm a physiologist, so I have to hire biomedical engineers in my laboratory, right? And then work with people like you, because I think that's the key now to this field, right? Is the different skill sets that people bring to the table. The quantitative skill sets, the qualitative skill sets, thinking outside the box in areas that you wouldn't normally think about, the things that we bring to each other that makes us think outside of our own box. And I think that's the fun part about science in general.
If you go back 50 years, would the medical community have envisioned that, the modern day DBS team includes a nice triad of a neurologist, a neurosurgeon, and a psychiatrist who employ many biomedical and aerospace engineers and and basic scientists and, a fleet of, research coordinators, it's just such a weird, amalgam of, a very diverse group that makes this happen. But definitely the medical model has not caught up with that.
What do you think they're going to say in 50 years? They're going to say, oh my God, they were sticking leads in people's brains? Why would they do that, right?
Right, without an ethicist at the front lines, even though we do have an ethicist, but still, it's just, it'd be hard to imagine, where things are and, the needs that we have now. Like, when we go into the OR and then I'm coding my case, I still can't code for the biomedical engineer. So at an academic institution, it requires so much more than if we made the decision to just do it based on imaging. And I think, that is sort of the hard bottom line that we're often fighting is really that tension between sort of the modern medical enterprise and, what we think is right.
Yeah. It's, when you think about neuroscience in general, we think about the brain. It is everybody talks about it's the next frontier, right? How does this thing work? How do you originate a thought?
Right.
And where does it come from? What makes us who we are? These are things that, maybe it depends what your beliefs are, right? Maybe when I die and go to heaven, hope we go to heaven, then maybe someone will tell me how this works.
I hope so.
I don't think I'm going to figure it out the time I have left on the earth, but I can at least make a little headway in some things, right? But the fact is, it really is kind of a very exciting area to think about it. And if you think about neuromodulation, you guys, I know you think about it all the time. how do we change people's behaviors and like mood disorders, the OCD world, depression world, schizophrenia. What's going on with schizophrenia? What are the circuits that are involved here? Where's this coming from? And is it multidimensional? There are multiple different spots. Like if you look at tics, you look at Tourette's and tics, and you wonder, could I affect tics by doing stimulation in different areas? I mean, the people that publish on that publish on I don't know how many different targets that they use for tics. So it's a distributed network, right? You might be able to get it at a lot of different places. Well, could we do something like that in Parkinson's? It is distributed to some degree, but the bottom line is it's pretty compact in STN and GPI. So we feel we can affect a lot of different circuits in a small area in STN. But if I go to the cortex, do I have to cover a broader area and do the same thing?
Circuit modulation, distributed circuit modulation.
There you go.
Yeah, that's what we're trying to do. So, Dr. Vitek, one of the things I think is most compelling about the Udall Center that you put together is how it's truly a translational project, right? It crosses from basic science to translational research all the way to clinical research. And I think, in my limited experience, that's a pretty unique entity. Seems to me it poses particular kinds of challenges to be able to put that together. And I was wondering if you could, talk a little bit about how you're able to have the vision for it and also work out the mechanics of making it work, because
Alex, I'll be honest, I think the key for all of that is people, right? It's just people. And you really have to have the right people that you can pull together. I mean, if you listen to what PJ Fleck says in the football team, he always talks about fit, right? You don't always just get the best athletes. You just want to get the best fit of the best athletes. And I think that's kind of the key. If you look at, for example, Matt Johnson in biomedical engineering. So Matt was at Cleveland Clinic, and then he came to Minnesota, and we did it about the same time. And Matt was asking, because he does non-human primate work, if I came, would we be able to build that lab, and would he be able to be a part of it? And of course, he's the consummate collaborator and very bright guy. And so what I was told many years ago by Malin is that just get the best people who can work together. And that was the concept. So when I was here, we talked about putting a Udall in early on, but there was another person in neuroscience who was going to put it in first. And I didn't want to, I thought they had the idea to put it in. I'm a newcomer in the block, but then put it in first. And they made a couple of attempts, and then they were not going to do it again. When they decided not to, I thought we need to go in. And the point then was, who do we pull together on this? And on each project, in order to do this, you have to have an R01 to be eligible to do a project in a Udall. So the bottom line was we had 3 people that I knew of with R01s that I thought would be complementary. And one of those was Matt, and Matt did non-human primate work, and he's very good at what he does. We were doing DBS at the time, so I thought, fine, I can do a project on the DBS side. And then Colin McKinnon, who does a lot of kinematics and studies circuits as well, I thought he was a natural for the third one, right? But there really weren't many more people that I could pull in. And I thought that made a nice group, because you have to, the projects there, right, the sum of the parts the projects have to be greater than the sum of the parts. So each one has to fill a slot that the other one can't, but yet they're still kind of in the same common thread of the question. So that formed who we had to bring in on this, right? And then how we put in our aims also had to be complementary. So that took some thought. And then you got to put core facilities in. Well, look, the things that I think are really strong here, we felt electrophysiology was one and imaging was the other. So naturally, it made sense that Morel would come in with an imaging core. And then we had to have administrative core, stats core, and then of course, because it's clinical work, it would be a clinical core. And that's how that got started. Then who are you going to have do those things? Well, Noel was a natural. I've been working with Noel. We had Scott Cooper and Paul Tewitt that made sense on the clinical core. Statistics, Lynn Everly, right? It made sense. And Eric Maurer, frankly, is the consummate administrator and clinical research person. So that made sense. Yeah. So we had the pieces, right? But then it was a matter of trying to pull it all together. I won't belabor all the details of all that, but we did a lot of thinking about, how to tie everything together well. And we decided, let's put it in and see what happens. And so we weren't sure. I didn't feel very confident the first time in because there were a lot of things we needed to do. But I thought we need to go in and let's see what kind of score we get. And, if we get funded, it'd be great. But if we don't know what we need to do. 2014, got our score back. We didn't get funded. We're like a 41 or something. And I thought, well, we can fix it, right? So let's take a look. We took 2 years before we went back in again. And then we went back in and we got funded on the second time in. But then, you have to think about, my approach is once you get it in 2016, now we need to be thinking about the renewal in 2020. And how are we going to do that? And, what's going to be new about the UDOL? And that's why I approached you guys on the cognitive phase, because I thought this has got to be the next area we move into. It's a natural evolution. It's also a major problem, right, that we know that the patients have. Mm-hmm. So I thought that would be exciting and new. And then there were other things that we got into, right, with the fiber pathways and the work that Matt was doing and things like that. But, really, Alex, for me, it was largely how we're going to organize the vision of the UDOL, but also the people we need to pull in. And because, this is a close group, right? I mean, we're working pretty closely together. And everybody has got to have some skin in the game. Everybody's got to feel that they have their own thing. Because that's what generates the juices, right? And makes you creative, makes you want to do this work. We have a lot of young investigators, right? In this group, we have 6, 7 young investigators, and
yeah.
But man, that brings in fresh ideas, new blood, enthusiasm. It just makes it a lot more fun.
Dr. Vitek, thank you so much for joining us today. It's a fantastic conversation. We really appreciate just being able to have sort of a casual conversation that others can listen to, because I think it's, in this day and age, it's quite rare. It really is rare to talk about this kind of stuff, the sort of overarching views and where things are heading and the excitement around it, because it's either clinical, where we're having sort of private conversations with patients, or technical, where we're talking with each other about very specific details, or grants. And there's just not a lot of venues to talk about the really cool long-term vision that you've had and what that's turning out to be. So thank you so much for coming.
Oh, my pleasure, you guys. You guys are doing a great job with this podcasting. It's really great to get this kind of information out for folks. And it kind of gives people a sense of where we've been, where we are and where we're hoping to go with this.
Absolutely.
And I just want to add, we're tremendously grateful for the opportunities we have to work in the UDOL. It's just the most exciting, work we're doing, and the vision and leadership you have for that program enables us to be able to do that work, and we wouldn't have these kinds of opportunities at other places. So thank you very much, and we're going to do our best to give back everything we can to it.
Oh, you guys are a blast to work with. I mean, this is fun for me. Malin always said, like I said, just get the best people and it will take care of itself. You guys are some of the best people.
Thank you so much, Dr. Vitek.
You're welcome, guys.